Good morning, students. I hope you all are fine. In my previous lesson, we discussed about vegetative propagation by underground stems, that is corn, rhizome, bulbs, and tubers. Now, in this video, we will understand about reproduction by leaves, roots, and one more method of vegetatively producing new plants, that is tissue culture. And one by one you will understand all these. First, reproduction by leaves. Now some plants like bryophyllum, they have large and fleshy leaves, and at the margin of these leaves several buds develop that grow roots and new leaves. Now these buds, they are capable of dropping off. They fall into the soil, they develop roots there, and new plant starts growing out of them. So in this way, these buds, in case of this bryophyllum leaf, is capable of producing new plants when it develops buds on the margin of the leaves. Sometimes what happens, the whole leaf may break off from the parent plant and develops root there, and this leads to development of a new plant. You can see here in this picture, this is a bryophyllum leaf, and you can see here these buds, they develop on the margin of the leaves in the notches present there. They are developing leaves here. They are capable of falling off into the soil. Now they develop roots there, and now they are capable of growing as independent plants. So this is reproduction by leaves. Next, it is reproduction by roots. Now some plants, they have fleshy and swollen roots. These are modified roots that store food. Fleshy and swollen means they are storing food inside them. Now these modified roots, they grow adventitious roots on them, which develop into new plants. Example, sweet potato, asparagus, dahlia, carrot, etc. So you can see here it is a sweet potato plant, and you can see these roots coming out of sweet potato, and when they are grown in the soil, they are capable of growing into a new independent plant. In dahlia, the roots need to be planted along with the buds to grow into independent plants. And in carrot plant, there are buds just above the swollen taproot. The carrot that you eat, that is a taproot, and just above that there are buds, and at the base of the stem there are buds in between, which are used for reproduction and developing into a new plant. So in this way, these roots in these plants are capable of producing new plants. Next, it is tissue culture. Now what is tissue culture? It is a method of producing plants in an artificial medium. Now what happens in this case? Few cells from the growing or other part of the plant can be taken. Few cells, and then they are kept in sterile medium which is rich in nutrients. Means all the nutrients which are required for the growth of plant, they are added into it, or the medium is selected in such a way that it contains all the nutrients inside it. Now these cells which are kept in the medium, they grow into small plantlets in a few days, which are then transferred into compost, and then they can be transferred into soil. And examples are orchids and asparagus. Now you can see here in this picture, there are these few cells which can be taken from the petiole of the leaf. Then these are transferred into a medium. The medium used here is agar, which is rich in nutrients, and few days you can see here these cells, they grow into small plantlets, which are then transferred into compost, and this compost provides other nutrients to it, and then these plants can be transferred into the soil to grow as independent plants. So this is tissue culture, in which the cells are grown in artificial medium and then transferred into the soil. Now what are the advantages of this method? It is a very fast method of growing the plants in very less time. Second, even if the plant is diseased, but if the cells are taken from the growing part, that is the tip of the stem or the tip of the root, then the new plants which are formed by this method, they are disease free. So this is the advantage of tissue culture. And one more advantage is large number of plants can be grown from a few cells. Large number of plants can be grown. So these are the advantages of tissue culture. And now it ends our topic here. We discussed here the asexual reproduction in plants. Earlier we discussed about various methods such as binary fission, then budding, fragmentation, spore formation, etc. Then you discussed about vegetative propagation through subaerial stems, underground stems, and in this video you understood about vegetative propagation by leaves, by roots, and artificially growing plants in a medium and then transferring into the soil, that is tissue culture. So we end our topic here. You can ask me your doubts in the time which is given to you on Telegram. Thank you, Vidam.